a break in between, so I was able to digest some of the things that were being spoken on, and then had enough time to get a nice little break, come back. So that I feel like that format was fantastic yesterday. So I hope you guys got a lot out of it, and I hope that you're able to digest this for the next 4 weeks here as we continue to improve our overall understanding of the soil food web and just cultivating cannabis in general using the microbial world. I wanted to also thank Dr. James White yesterday, as well as Chris Trump and Leighton Morrison for giving us fantastic presentations. We also had Daniel as the host yesterday, so thank you to Mr. N. King. Today we have Dr. Efren-Cazares, who also understands the microbial world and specializes in the mycorrhizal fungi. So we're going into a deep dive into the mycorrhizal fungi world. And this is something that I know that when I started to read a lot of different books out there, this is kind of a game changer, if you will, especially when you start to understand that it's not really necessary until you start building those living soil systems, until you start building those raised beds. So I want to also give a shout out to our title sponsor, Agro Coco. Please check them out. It's at Agro Coco on Instagram, as well as our session sponsor today, sponsoring Mr. Efren's talk. That is from Pit Moss. So check them out at Pit Moss Official on Instagram as well. Mr. Efren, I will give you the board there, and thank you, Daniel, as well, if you have anything else to say, sir. All right.
No,
thank you, Agriflex Universities, for putting this on behind the scenes. There's a lot of work that it takes to put all of this together. So thank you to everybody involved on that as well. Mr. Efren, you have the show, sir.
Thank you. Good morning, everybody. See about 75 people attendees. Good afternoon in the East Coast in Australia. I think I heard somebody was in Australia. Anyway, so thank you for the invitation to Leighton, Daniel, and Brian. I was able to see and watch the presentations yesterday.
Excellent.
And very good presentations, except Leighton's though. He's all right.
I like that you give him shit, man. Seems like you guys like really old school friends, so that's good to see.
So I'm gonna talk about mainly mycorrhizae, but I'm gonna give you a very quick introduction to fungi, to mycology. It's literally 101, because mycorrhizae is a very special field and a lot of the terminology. So I'm going to overwhelm you with information and terms and concepts, and but I would try to make it fun. So that's the first section, is introducing the concepts. And then I'm going to talk a little bit about the ecology, what they do. And then I'm going to define the different types of mycorrhizae. And then we'll go back again to some of their ecological concepts and what they do. And then we can pause somewhere in there and take a breather, and people can ask some questions. And then I'll go back again, and we'll talk about some of the stuff. And then we end up with a little bit of cannabis and mycorrhizae. So
here, let me start that for you.
Yep.
All right, and then I'm gonna fall off until you're ready for questions, sir.
Okay. So to begin a presentation like this, I always like to show this slide because these are the two main chemical reactions in the most important chemical reactions in this earth. They're the driving force, and this is what feed the rest of the trophic levels, and anything. So photosynthesis, sunlight, CO2, and water. Water comes in, goes into the plants, and we get photosynthesis. So we're creating carbohydrates, and that's where the energy is stored for the rest of the organisms. Those carbon bonds, those little sticks in the chemical formula, when they break, they release the energy, and that's what everybody is after. Now, respiration is not the inhaling, exhaling, and inhaling. It's about the metabolic respiration. It's the opposite reaction to photosynthesis. When you break the sugars, you release the energy and produce CO2. So those are the main reactions. And the other thing that I like to highlight is the enzymes. All enzymes are proteins, pretty much. Most of the enzymes are proteins, but not all proteins are enzymes. And so every organism had to produce those enzymes to break down the big molecules so the molecules can go through their bodies, in a simple compounds. So enzymes are really, really important for the ecosystems. So fungi. In the old days, it used to be like plant animals and minerals. Even now we know at least there's recognized 6 kingdoms, and it depends on how the nuclei are in their cells. They have membrane or not. But fungi is their own kingdom. They're up there, and so they have their own thing. They're not plants. A lot of people think they're plants. Let's define. Let's talk about fungi. So, because I'm a mycologist, I like to say that they're the most important organisms in the world, and because I said so, they play vital roles in ecosystem functions. Humans, we depend on fungi, even though we don't know, but they're everywhere, and they're doing a lot of functions in the ecosystem, mainly decomposing, nutrient cycling, and nutrient transport. We'll talk about the numbers in the next slide, but their estimates, they go from 1.5 to 5.1 million species of fungi. That's the estimate as present existing organisms. But only about 100,000 species have been described of all fungi. So just imagine the work needs to be done to discover and give names, and not just name, but their functions too. So we know, from beer making, wine, medicines, food, you name it. So let's define fungi. They're very diverse, and they don't make energy. They have to consume it, just like us. They have to go after the carbon compounds. In their body are microscopic filaments, and they call them hyphae. That's the filament, microscopic filaments. They have to be in direct contact with their nutrients in the environment. They produce extracellular enzymes. And as I was saying, the enzymes have to break down this compound so they can go through the cell wall. They don't have a mouth. They have to go through the cell wall. And then they can survive in pretty much every substrate. You can find them in many, many places, but they have to be an intimate relationship with their substrate. The numbers, I was saying, there's estimates, some conservative estimates of 1.5 million species to 5.1 million species. 5.1 million species. And this is based on a lot of DNA studies. But just imagine that it takes about 1,500 species are described per year in average. Imagine how long it's going to take to describe 5.1 million species, more than 3,000 years. So it's a lot to be done, and we assume that a lot of the functions that are doing in the soil, we always blame it on particular species, but unless you have controlled experiments, you just don't know who is causing the effect. When we talk about plants, there's about 400,000 species of plants, but they're divided. Most of them are angiosperms and so on. But out of those plants, there's about 3% of the plants known are known to have mycorrhizal hosts and have been studied to have mycorrhizal. There are mycorrhizal hosts. When it comes to fungi, within all those fungi, we only know about 6,000 mycorrhizal species. And out of those 6,000, we have about 300 arbuscular mycorrhizal fungi described, and the ones that we're going to be talking about. And specifically, for example, Douglas fir, a common tree in the West, is known to associate at least 2,000 mycorrhizal species, specifically to Xerozuga. So there's a lot of numbers to digest. So I'm going to show a lot of pictures. The hyphae, when they're together as an individual, all those group of hyphae become mycelia. And this is the mycelia, and the mycelia, it goes and surround the substrate, and that's because that's what they need to be attached to it so they can eat. You can see the rhizomorphs on the left bottom left is an ectomycorrhizal tip. How do they disperse this fungi? Well, they produce all kinds of pretty spores. Nobody knows why the ornamentation is so diverse, but a lot of the spores are produced by are dispersed by air, some by water, and some others are consumed by animals, and that's how they get dispersed. On the fungi, most of the fungi that we know belong to three different classes or subdivisions. The ascomycetes, or the cup fungus. Those are the spores are developed inside of a sac. It's called asci, and they're inside, and that makes it a totally different fungi compared to the basidiomycetes. The basidiomycetes, a lot of the mushrooms that we know produce the spores on top of a structures called basidia, and usually are 4, but sometimes 6 or 8. It can vary, but mostly are 4. And so they're produced on gills, on tubes, enclosed on a fruiting body, like a protected by peridia, a layer, and so they disperse again by air or consumed by animals. Now, any fungi that you can think of is going to fall in one of these categories: decomposers, parasites, and the mutualists. It doesn't matter what fungi is going to be in these categories. This is what they do. Decompose the organic matter. That's how they get their nutrients. Parasite, they have to be in a living organism. It's also a symbiosis, but it's not a mutualistic symbiosis. It's a parasitism. And then you got the mutualists with both benefit. And then you got the endophytes that Dr. White talked about it yesterday, and some of the lichens, they mix with an algae, make a lichen. And then the mycorrhizal fungi, and those are the ones we're going to be focusing. So let's define mycorrhizae first. It's a combined word: fungus, mycofungus, and rhiza root. And that concept was developed in the 1800s, late 1800s. It was thought to be a parasite, but it ended up being a mutualistic symbiosis. The easiest simple definition is that it's just an association, a mutualistic positive association between a soil fungi and plants, and this through the roots. A more sophisticated definition is that this relationship is defined because the fungus obtains minerals and water where the plant cannot reach because of the threads, the microscopic threads, compared to the diameter of the root. And so they associate with the root, and it passes those nutrients and the water in exchange for the energy, the carbon sources, the carbon compounds that, they're obviously photosynthetically derived. And also in the diagram on the top, it's a three-way interaction. You have to have a plant with roots. You have to have a substrate, the soil, and you need to have a fungus that provides this type of function, a mycorrhizal fungi. When you got those three things together, that's when you form a mycorrhiza. Now, this is not a new, I mean, obviously, it's not the mycorrhizae concept. It's new, but they've been with the plants since they migrated to Earth. 400 million years ago. They already evidence that the fungi were already associated with plant-looking things, photosynthetic. They already needed, the fungi already needed to associate to get the carbon from somewhere. The estimates are that about 80 to 90% of the plant species are mycorrhizal hosts. So they need that association. In general, all conifers, cycads, they're all mycorrhizal hosts. There are about 6,500 plant species that have been examined for mycorrhizae. Remember, there are about thousands of species that are known, but only about 3% have been studied and examined for their association, and mostly agricultural crops for obvious reasons. So there's a lot to be done. It's just a diagram just to show you that, when did the mycorrhizae show up in the Devonian? When we have an Earth at 4 billion years old, but the mycorrhizae shows up in the Devonian. What are the benefits of the mycorrhizal fungi? And this is in general, this is generalities based on thousands of studies, about 30,000 studies of publications on mycorrhizal fungi. The main one, as I mentioned before, is that they enhance the nutrient uptake. And why is it enhancing? Well, if you think about it, the size of a fungal hyphae in general, it can be 1-2 microns in diameter, 5-10 microns, can be 40-50 microns in diameter. But the roots hairs, or the roots, can be 1 millimeter, 2 millimeters. But 1 millimeter has 1,000 microns. So just think about it, how many hyphae you can put in the same cylinder when it measures 10 microns on 1,000 microns. So the hyphae explores areas where the roots cannot go to. And so the micropores and places where the water might be in storage and the nutrients. And because of the same reason, they enhance the water uptake. And therefore also they provide drought resistance because of the association with the fungus. In many cases, the fungus produces a mass of hyphae, and that kind of puts a barrier, a mechanical barrier against pathogens. They promote nitrogen fixation. They increase root longevity because it just makes it stronger roots. And a lot of the fungi produce fruiting bodies. They're food for animals, which they just keep providing energy for others, nutrients for other animals, organisms. When we hear mycorrhizae types, there's a quite different associations in how the fungi attaches to the root and what it does once it gets into the root system. So many of you probably heard of the term Ectomycorrhizae, and we'll find the types. We have the Arbuscular, which is the most known and abundant. We got the Arikoi with Orchideoe with orchids, and Ectendo is a combination. Arbutoi with different, specific to Arbutus family, monotropes. And then we got the dark sapendophytes, Dr. White talked about them. I did a lot of assessment and study with this during my dissertation about 30 years ago. And so we keep finding these dark sapendophytes in the root systems. So basically, the types are how the structure, how the fungus goes into the cortex cells of the roots. The Ectomycorrhizae, as to I don't have a pointer. The Ectomycorrhizae, the fungal hyphae, these filaments surround the cortical cells, but do not penetrate the cell. They just stay around. That's what they call ecto, outside, external. And when they do that, mainly with trees, when they do that, they make a barrier, and you'll see some more pictures. They put up a barrier outside, kind of changes the morphology of the root tip. And then we got the arbuscular. It used to be called vesicular arbuscular. Now it's only called arbuscular mycorrhizal fungi, and that type goes inside the root cells, and I'll show you better pictures. This is just a diagram just for comparison. It goes inside the cell, not exactly in the cytoplasm, but there's a membrane, plasmalemma. It looks like they're inside completely, but they're not. And then we got the Arikoi, totally different fungi. They make this spaghetti-like stuff. It's totally different fungi, so it's just a diagram to see the differences. Let's show the Ectomycorrhizae. As I say, the hyphae surrounding on the top right is a cross section of a root. Those little round things is a cross section of the hyphae, making a layer. They call them mantle. And then you have the hyphae surrounding the cortical cell. They call the Harding net, and it's just like a really tight interface with the root, and that's where the nutrient exchange is happening. And what fungi produce? A lot of mushrooms, a lot of truffles produce this type of mycorrhizal type. What plant host? Most of the conifers. The Pinaceae family, Douglas firs, spruce, pines, hemlocks, eucalyptus, some legumes, acacias, and others, even carrias and pecans. And so they changed the morphology of the root tip, given different colors. It's rather beautiful to see them under the scope with those different. And the fungi, it can be. I've seen seedlings, 2-year-old seedlings of Douglas fir with about 25 different types of mycorrhiza types on them. So it's amazing to see them under the scope. What are the fungi that produce this, that make this association? A lot of the mushrooms, and you have to remember, the mushrooms is like an apple to a tree. This is just the fruiting body. The individual is attached to the roots, and the soil attached to the roots. And when the conditions, seasonality, moisture, and temperature are favorable, they form these fruiting bodies, and this is how they get the spread of their spores, and to continue colonizing substrates. Then we have the Arikoi Mycorrhizae. They don't produce a mantle; it's just fuzz outside the root, and no Harding net, no mantle. It's just coils inside the root cells. You can see in the middle of the brown slide, they make spaghetti things, and it just, they go from one cell to another on the outside, not through the middle. You can see in the bottom picture, how the hyphae goes outside the cell and colonize another cell. And this is only specific to the Ericaceae family. Like blueberries, azaleas, rhododendrons. Really, really interesting type of mycorrhiza. And then you got the arbuscular mycorrhiza, the most common one. This is the one we're going to focus because that's the one that is considered to colonize or to form it with cannabis. So no mantle, no none of that stuff. They make this hyphae coils inside, and the arbuscules, which is like the bottom one, is like a 3-branch type, and it looks like a little fuzz. And because what happened is that the cell wall of the fungi, for example, it goes from 1 micron in thickness, and when it forms the arbuscule, outside the main hyphae, the arbuscule, the thick wall, goes from 1 micron to a nanometer. So the cell wall keeps going smaller and smaller. And And what happens there? Well, if it facilitates the nutrient transfer between the plant and the fungus, it increases the uptake area, the transfer area by branching, increases the surface area tremendously. And so, as I say, most crops, shrubs, forests, herbs, a lot of trees, pretty much any plant except the Brassicaceae family, the mustard, the broccoli, the cabbages, they don't have mycorrhizae at all. We don't know why. Maybe some chemicals are stopping. Now, the importance of the arbuscular mycorrhizal fungi in the soil, they're known to have, they're the only one that produce the glomalin. The glomalin is the glycoprotein, it's the soil superglue. They say it's about a third of the soil, carbon is by glomalin. This glomalin, this glue, aggregates the soil, and it just makes it stable, stabilizes the soil, and that's where a lot of the nutrient uptake is possible. And this aggregate takes a water holding capacity, increases a lot of water holding capacity. The middle picture shows, spores and hyphae. It's not actually the glomalin, but the glomalin is a glycoprotein, so you can't see it. So next. Now let's talk about the fungal life cycle. It's rather simple. You got the hyphae, it's a fungal individual. And then you got the spores. They're produced by the fungal hyphae once they go inside the cell. And it creates all the structure, the vesicle, the arbuscules. And then when it's time, they produce the spores. The spores are single spores in the soil. Sometimes they're produced inside the roots. And like the most common one, Globus interradices or Rhizophilus interradices. So that's it. This is the cycle, and this cycle happens. It could happen 3, 4, 5 months, and the species is done. Obviously, not all species do it at the same time. And so these are the propagules, the hyphae, the spores, and the mycorrhizal root fragments. When we talk about mycorrhizal fungi and arbuscular mycorrhizal fungi, we say 200, 300 species, but there are very few genera compared to other fungi. And the ones with the asterisk are the ones that use commercially. They're produced commercially because they produce a lot of spores and they grow quickly. And I'll show you a lot of the spores later. What are the structures? As I said, the arbuscules, they're very branched, very fine structures. And then we got the vesicles, not all produce vesicles, but they got the vesicles going. And different shapes. Now, to see this, you have to clear the roots, take all the pigments with some chemicals, and then stain the roots, and they'll stain the fungus, they'll stain the cell wall. That's how you can see it. Otherwise, you can't see them unless you stain them. So here, again, most of the mycorrhizal structures. And then we're going to show some of the spores. The spores, these guys don't form mushrooms, right? They produce the spores in the soil. The spores are single. They just go there. Every spore you find in the soil has a potential of a different name, and that makes it complicated when you extract spores from the soil because their taxonomy is based on a single spore, and it's just basically size, color, cell wall thickness, the structure, how the hyphae is attached at the bottom, and that's it. That's how they distinguish their species names, the species. These are some of the pictures I've taken through the years when you extract spores from the soil, and I'll go through the method too. Very diverse. You can see a lot of diversity here in the middle. The middle pictures on the top. The famous Glomus mosi is on the top, the big spore with the funnel-like thing. It's called. It used to be called Glomus. Now it's called Funneliformis, a funnel-shaped mossy. I isolated this. the hyaline, the no color ones on the right and the middle, those were isolated potted plants. And it's just a beautiful diversity of spores. As I said, to put names on them, you have to study single spore only. Here's another more pictures of the spores, single spores. It's just little balloons. They measure about 40, 50 microns in average. You can go up to 100 microns. Remember, 1 millimeter is 1,000 microns, just for reference. Recently, I was doing an assessment on some grasses, and this I spotted. I've never seen this before. I spotted a bunch of spore being produced, but if you see the inside, there's a fungal endophyte or a parasite growing inside the spore. Brown hyphae inside the spore. I couldn't resist to show it to you, and I never seen it before. But anyway, so in commercial inocula, I don't know if you use commercial inocula. They use the term propagules, Arbuscular mycorrhizal propagules. What they're doing is the propagules, again, is spores, mycorrhizal fungal hyphae, and root fragments colonized by mycorrhizal fungi. Any of those propagules can potentially generate another individual. And so when they sell you a bag with mycorrhizal fungi in it, it could have the three of them. The problem with the numbers that they use is that they give you a total number of propagules. And it's kind of fussy because they don't tell you the proportion of each of them. So that's why the numbers are really high. But on the right-hand side, you see a couple cells came off from the root easily, and they're already colonized. So that is a potential propagule. So in the middle picture, you see a little flake of vermiculite. The spores grow inside the vermiculite layers, between the layers of vermiculite. It's pretty cool to see when you stain them. More pictures of propagules. On the right-hand side is mycorrhizal fungi produced in vitro. In the middle is in vitro. The one on the right up top is called Rhizophagus aggregatum. You wonder why it's called aggregatum. The aggregate. And then you got root fragments with spores on the left. And then you got a citrus fragment colonized, and obviously the vesicles starting to turn into spores themselves and produce a root fragment full of spores. These fragments come off and the spores get, released in the soil. That's Glomus intraradices or Rhizophagus intraradices. More pictures of propagules, scanning electron microscopy, showing abundance of hyphae. Now the top right picture shows the same species with the different sizes and development stages. So imagine if you put one spore outside of this conglomeration, how do you put a name on it? That's the difficulty of this, of trying to put names on single spores, because it could be anything. And so not easy to identify. And then you got some root cells colonized with the mycorrhizal fungi, and a lot of spores on the bottom with different tones of color. Now, remember we talk about the uptake of nutrients. The main and the most common in study is phosphorus. So phosphorus, the phosphorus is really important for the plant, and so this is where the fungus takes action. The phosphorus has to be in close contact with the root. The closer, the better. And so, how do you do that if the phosphorus is not reachable? Well, that's where the fungi comes in. And remember, the enzymes play a big part. You see, there's a representation. That's the the phosphorus depletion is just around where the root zones, the root tips and the root hairs, or if it's colonized by mycorrhizae, around the hyphae. Remember, phosphorus has low mobility and usually is absorbed as orthophosphate. It has to be broken down. It's not accessible. So how do you break it down? Well, the fungus produce phosphatases, enzymes that break down these compounds and make it available for the plant. Otherwise, will not be able to penetrate, through the cell walls. So they have to be breaking down and make it a lot simpler. Just a diagram to show the extension and the extension where the fungal hyphae can explore a lot of parts of the soil. So let's talk about a little bit of the ecology. Disturbance is really interesting concept and subject when it comes to soil management or what happened to the organisms. It takes a lot of years, a lot of time, a lot of interactions, a lot of organisms to become stable in the soil. You go into your native plant communities and they're doing whatever they do, no human interaction. But disturbance comes in when you bring agricultural systems, and the same management over and over obviously damage the structure of and the harmony of the soil ecosystem and all the organisms that are living there in harmony. So you disturb the soil and the organisms that don't know what the heck happened. What do we go? What happened? We were nicely, eating nutrients here and passing, and everybody's doing their job. And so, how do we put this disturbance? Is it a one-time event, or is it a constantly happen? Every year, every 6 months. Agricultural systems, they keep moving the soil, so they keep making things worse, and you have to remediate immediately so you can have a crop growing for the next season. So when that happened, a lot of depletion, compaction, pollution, flooding, and the populations fluctuate. Some increase, some decrease, others just disappear. They can't live on stable conditions. So the diversity fluctuates. Decomposers, symbionts, pathogens, opportunistic, and so it modifies the soil structure, the water holding capacity, and obviously the productivity changes. And just for a little parenthesis, when you buy soil in a bag, it's all disturbed. To me, it's disturbed soil, right? You put it in a pot and you try to grow a plant quickly. So you put all these inputs, amendments, and things trying to fake, the system for the cannabis plant or whatever you grow on a plant, on a plot. And so that's what all these amendments and recipes and things trying to replace, the environment. So think about the soil is a disturbed soil, and so anything positive that comes out of that disturbed soil is basically a miracle. The plant is taking whatever you can give in any shape or form. Easy or very hard to get, and you have very few months to do that because the plant needs to grow, and you need to harvest and sell the little nuggets. So what are the practices? Well, we all know, you remove the organic matter, the chemical fertilization, the compaction changes all the water systems, how the water flows when you compact the soil, big areas of fumigation, the tilling, the erosion, everything is changing below ground. All the organisms are in a chaotic state. And so it makes it really, it's a miracle at how some of the microorganisms can come back on an agricultural system. It's just a list of, I get questions of what fungicides affect the mycorrhizal fungi. And a lot of them I never worked with fungicides, but this is just a list, and this is some papers published. Some of the list on the left shows the ones that don't have any effect, and then some that have some effect at a low rate, and there's others that at any rate they can definitely deplete or stop the mycorrhizal fungi. This is a famous picture. What I like to say on this famous picture is that this is a very simplistic diagram. If in real life, I would think that we can put thousands of lines going everywhere, back and forth. But I just want to highlight the fungi because that's what I'm talking about. So the fungi have to be with the plants, and they have to do their thing, and somebody, some animal, some bacteria have to be involved in the root surface and passes all those nutrients to higher trophic levels. But the fungi are there, and everybody's, some of them produce enzymes to keep breaking down the compounds. And so I'm using this picture to pause a little and maybe take some questions, so I don't blow you with information.
Yes, sir. They're starting to come in, so we have a few that we could get started with. This one actually came from Instagram before things started. This would be the best way or best method to add enzymes when you're building a living soil.
I'm the wrong person to ask that. Enzymes. That's not my specialty. I think that was more for Leighton, I think, wasn't it? I mean, I don't.
Yeah, so I think they were acting. Yeah,
I don't know what type of enzyme they're referring to.
Yes, sir. All right.
And what would be the objective of adding enzymes? I'm trying to link it to mycorrhizal fungi.
Yeah, so I think. I'm speculating here, but I know like with sprouted seed teas and that kind of thing, are you adding enzymes if you're creating those kind of things?
No.
Okay. All right, this one is from Sean Russell. At what rate do we discover new fungal species?
Well, it depends on the groups of mycology on the world. Is that what they mean? Like how many species are published?
I'm just reading these off, sir, so I apologize if they're not clear.
I'm trying to see at what rate. Well, it depends on the mycological groups or people that actually work on taxonomy, because not all mycologists work with taxonomy. Some are very specialized. I started pretty deep with taxonomy, and then I move up, and work more on ecology on top of the taxonomy. But no, I don't know, but it takes a lot of time to describe species and get them published on top. It all depends on, I guess it depends on the mycology number, the number of mycologists out there, actively describing species, I guess.
Yes, sir. I appreciate that. And Leighton just chimed in that he'll be able to answer that. So thank you, Leighton. This next one comes from Joseph Pritchett. Have there been any studies showing at what rates of nutrient concentrations in soil do mycorrhizals stop forming relationships with plants?
Oh, yeah. That's a good one. I was going to mention that later. So we know that the two main minerals, phosphorus and nitrogen. And as a rule of thumb, when we find the best level for phosphorus in the soil for mycorrhizae to thrive is between 20 and 50 parts per million. And it goes about the same with nitrogen. Anything above, it suppresses the mycorrhizal fungi development in the soil. It's sort of like there's too much phosphorus, that the plant doesn't it takes the phosphorus that is given to the plant themselves instead of using the fungi. And so it's between 20 and 50 parts per million. And I'll show some data that Leighton gave me at the end to show that. But that, yeah, between 20 and 50 parts per million.
Yes, sir. This next question comes from Clay Whitcomb. Chelators can be plant hormones. So what is that phosphate that's chelated with IAA? I believe he's talking about the auxins there.
I guess I don't know. I'm not sure if I got the question.
Chelators can be plant hormones. So what is that phosphate that's chelated with IAA?
I think I'm missing something in there.
All right, Clay, if you could kind of define that, sir. There's one from Tomas. Would IMO collection or highly fungal forest duff from JMS contain mycorrhizal spores?
Very likely, yep. Very likely. Especially if they were mycorrhizal hosts living in that soil. Now, we have to remember, if you get forest soil and there's abundance of ectomycorrhizal fungi in there, maybe hyphae, maybe spores, those will not be able to propagate on a different area because they're specific. For them to propagate, they need to connect to the roots. It's not a decomposer that can clone themselves and provide more spores. If we wanted to propagate the ectos, you need to put a plant. If we need to propagate the arbuscular, you need to put a plant on top. You're only going to be able to propagate the decomposers, not the mycorrhizal fungi. But they're there, but they will not grow.
This next one is from Keith Nelson. Is it possible to grow wild evergreen in a cannabis bed outdoors? Would that bring in varieties of mycorrhizae? Would they keep soil alive during a winter cycle?
In theory, yes. Yes. And we'll talk more about it. Assuming that the fungi connect with the cannabis, yes. But we don't know until we check. And a lot of times, cannabis are so precious. Each individual is so precious that a lot of people don't want to touch them. They want to touch the roots. They don't want to damage the roots because it may affect their productivity. And so it makes it, you have to say, okay, you need to sacrifice some plants. So we need to keep monitoring a particular individual for how the mycorrhizae gets developed and follow the development, if any. So.
This one is from Gustavo. Is there any competition between mycorrhizae and bacteria on a living soil indoor environment?
I don't think so. I don't think so. I mean, if they're going for the same nutrient, yeah, maybe, but I don't think it's a problem. I think they're more beneficial when they're together because they're tapping on different sources. And now I have to take advantage. I don't know, when you have imagine a root system, let's say corn. You pull a corn and you got all this mass of roots coming from the soil. And if we think that there's mycorrhizal fungi attached to all those corn roots, all those roots, the distribution of the mycorrhizal fungi around on the root system, it can be very patchy. It's not 100% colonized. And that's why we do assessments. So it depends on where do you sample the roots, because you only need a handful of roots, to assess colonization practically in a practical way. So we cannot assess the 100% of the root system because it will take you forever. But if we sample an area, you may hit the spot where the colonization is happening. If I take two samples and then I have a replication of the number, so the more samples you have from one big root system, the more solid is the data. They're not bacteria where you can just take a soil solution and make numbers. This sampling is very important for mycorrhizal assessments, and it can get costly and it may not be practical, but you have to realize what one sample means when you're talking about hundreds of meters of roots, but you only assess 100 centimeters. So.
All right, I have a gold nugget question that I think a lot of viewers should write down here when you give this answer. It's from Leighton Morrison. And what's the difference in vitality between vivo versus in vitro produced propagules?
Oh boy, he's getting me.
Here we go.
He's getting in trouble. This is my opinion.
All right.
I work with mycorrhizae for 30 years already. And this is my assumption, my hypothesis. I work with in vitro and I work with in vivo as a consultant. Think about the in vivo. The in vivo is basically what the most common way to produce mycorrhizal fungi. And the process is, the commercial producers, the process is this. You have a pot, a bag, I don't know, about 2 feet tall, 3 feet tall, it varies. And you can put 1,000 pounds or 500 pounds of substrate, and usually it's expanded clay, and we'll show some expanded clay, vermiculite mix, very little organic matter. And you put the spores, and you grow a plant. And this is outside. Most people do it outside, in rows of these plants. They grow corn or sorghum or sunflower, vetch, things like that. And you're exposing the system outside. And so the fungi is already adjusting to the outside environment. It's not soil, but it's outside environment. Temperature, other organisms, is not going to be sterile because it's outside. And so they're hardier to me. They're already adjusted to the outside. In vitro, what they do is they use modified carrot roots obtained from tissue culture, and they before they do that, what they did is that they slice a circle of carrot, and they put a bacterium, agrobacterium, and I believe it's tumefaciens, and so this bacteria causes some kind of a deformation or induces the carrot cells to produce very fine hairs on the media. This is in tissue culture. And so they produce these hairs. And then once their hairs are clean, they cut them. And these hairs can be transferred to another jar or petri dish. And they can produce a lot of hair. They're called hairy roots. It's just hairs of roots in the media. And then they put a single sterilized spore. But, they wash it and clean it. And then they put the spore attached to the media where the roots are. And the spore germinates and the hyphae grows inside the carrot root. And then the root keeps growing, and the fungus keeps growing into the media in a sterile condition, in a jar or in a petri dish. And they make mycorrhizal colonization in the petri dish. It forms all the structures, and they keep growing. And then it comes to after a few months, they start producing spores. Boom. And it's likely because the nutrients in the system are being depleted. It's just the colony is getting older and older. And then they produce the spores into the media, either liquid or semi-liquid or solidified agar. Those spores that media is blended, and then they make this solution, a lot of spores and a lot of hyphae and a lot of root fragments. Now you got an in vitro inoculant, and then they dilute it in sterile water and make it a bigger volume, and that's what they sell. And I don't know how common it is anyway.
Do you think that gets weaker and weaker over time?
That's one. That was my point. That
right.
They keep cloning this. They keep cloning. They make a lot of cultures. So what is the difference between that in vitro versus in vivo? To me, is that this fungus has no idea what it's like to live in the ghetto, in the soil, in the middle of the jungle.
Right. It's rough out there in those streets.
Yeah, because they're gonna go into the actual living soil with there's a lot of things happening already, in the dark alleys and the bars and smoky rooms, and they're just, and they don't know what to do. They will not like, okay, how do I get in? I'm too clean. I can't get dirty. I know I was fed with the silver spoon. All the nutrients were there. I didn't know how to look for them. I just give it to me. So to me, physiologically, you're losing all the traits that you form when you were outside. And so to me, I think they're a lot weaker. That's my opinion, but I can be wrong. So the only way to test it is to set up a statistical experiment with a lot of replication and see. But, once you put them in the soil, it's very difficult to track this down as an experiment, because you're not going to have a sterile soil outside. And how are you going to know which one is the one that you put in there versus the native mycorrhizal fungi? So it's rather difficult experiment to set up. The in vivo, they're already outside. They grow with the plant, a regular plant. They even though it's not real soil, it's expanded clay substrate, but it's outside with the real plant. That is exposed to a competition and from different organisms in the soil. A lot of the commercial inoculants, there's a lot of other fungi, not just mycorrhizal fungi. I can see them when you extract them. There's a lot of particles of other fungi, brown hyphae fungi. They're not mycorrhizal. They're parasites, they're decomposers, they're maybe pathogens. Who knows? But they're not sterile. A lot of the mycorrhizal products are not sterile. So, but it's not going to hurt. When we talk about plant pathology, that you get all kinds of systems.
I think that was those questions are probably also for more of our own amusement, just to hear your answer on that. So I appreciate that, sir.
He put me on the spot, that little.
Yeah, he loves to do that, huh? All right, John Motis, I hope I'm pronouncing that right. When we see the Propagils program on a package, the Propagils, I think is right, Propagils program on a package of mycorrhizae product, a lot of the times we also see trichoderma and other things. What product would you recommend or products?
Yeah, unfortunately I can't recommend a specific brand, because I'll get in trouble, because I usually work with pretty much all the companies. And so I can't, it'll be too public, but I mean, all products are good. They have good intentions. They have nice labels, but the problem is, when you produce commercial inocula, the most challenging thing is to homogenize the final product that puts in the bag. I mean, I'm a consultant for pretty much all these companies, so I know how they make the product. A lot of secrets and all that stuff, But the challenge, and I also do assessments for the Oregon Department of Agriculture. I do blind tests for them. So they check in their products, but I don't know what products are. They just send me a sample. So I get the results back, and then they are in charge of which product it is. But even then, they buy the product off the shelf. The ODA, the Oregon Department of Agriculture, they get the products out of the shelf, and then they relabel it, and then they send it to me. Did a couple of years ago in a row. But the original pot where the inocula was produced, they need to harvest this thing. They grind all the roots, and the spores are not bacteria. They go in different spots, and then they got a machine and fill all the bags. Now, to have a strong quality control, you need to know how many spores are going in each bag. Is it on the same proportion? So homogenization. So when somebody buys a package or a bottle, they need to shake this thing on top of it and mix it all up. So to have an even thing, that's just one thing. What says on the label may not be what's actually in there. And right. So that's the big issue for a lot of companies, even though they don't do it on purpose. It's just the system, how they work. So I have this issue with a few clients that says, my product doesn't seem to be working. And they send me samples, and I say, well, I have no idea what the product looks like or what's in it. Do you have a sample? You still have the product? So why use it all? So you can't do anything. I said, well, I got some left. Well, hopefully what was left is representative of the whole package. And so you would do extraction, and you find half of it, what the label says, or a quarter. And so your dilutions don't make anything, because you think you're adding 100 spores per gram, but in fact you're adding 20 spores per gram. And if you dilute it, well, you're not diluting 100, you're diluting 20, but you don't know. And so it makes it very challenging for everybody, for that particular part. I hope somebody asks me. I don't know if they're going to ask this question. Maybe that this will help. Some people say, okay, so another thing, the Arbuscular Mycorrhizal spores are dormant by nature. When they get produced, they are dormant almost immediately. Their cell wall is really thick, as you saw. And they're resistant spores. They're chlamydospores. They're resistant spores. They're supposed to stay on the soil until they're ready to germinate. And that spore has nutrients, and the package is there. So when it germinates, it has enough energy storage in the spore to keep growing with that the plant root. Because the spore germinates, and if there's no root available, of a host that can take him in without any issues, the fungal die. The individual die. But if he finds a root that is that will take a fungal symbiome, then it colonizes this, and then it's done. He was lucky. And so the more spores you have in your substrate, the bigger the chances. Now you're forcing them to grow in cannabis, for example. And so if the spores are not ready to germinate from your package, you only got 3 or 4 months to get them growing, because the cannabis grows really fast. And so my hypothesis is that people put the inocula at the same time as you put, the plants are starting to grow, but they're growing at a different rate. The plant grows too quick, and the fungus is still sleeping. And the plant is producing a lot of roots, and the fungus is, who's moving me? I'm still sleeping. And so by the time the fungus germinates, the plant is already, full adult. And so I think synchronizing the germination with the roots is a key for me for cannabis. And one way is to, how do you break dormancy on a commercial product? My suggestion is, and I don't know how, how everybody has their own ways to prepare their soil. My suggestion is, one is to put your inocula in the freezer or in the fridge for a month before you use it. And so you're breaking dormancy using cold temperatures, just like they do it in nature in the temperate zones. The soils get cold, right? Some froze, get snow, cold temperatures, cold temperatures, cold temperatures. And the fungi will come back in the spring like nothing happened. How do they survive? How do those spores germinate? Well, they broke dormancy by seasonality, by the conditions of the environment. Well, let's mimic those things and put them in the fridge before you use them. Other is to apply the spores to the soil before you're even going to use it. So mix up your inoculants and just let them sit there, until they do their thing. You're going to have plenty, and then you put the plants, so when they're germinate, the fungi are already ahead. It's about to wake up by the time the root's starting to produce. And so there's some kind of a synchronization there. I think it's a timing, the application of the inoculants. I wanted to mention that. I think I talked to Leighton before about this. So, any other things?
I think, especially for newer farmers out there, how you had mentioned, setting things up, being proactive. A lot of out there, I see a lot of people are missing that. So I like that you highlighted that. I also wanted to know, are you familiar with there was a report basically done from the Department of Ag, Oregon Department of Ag, where they were doing research on certain products, microbial products. That was probably about 4 or 5 years ago. Have you any recollection of that?
Actually, it must have been me, but it's not about, I think it's just the testing, at least when it comes to mycorrhizal.
It was kind of controversial for some of the brands, and that's why I was wondering if you had an opinion on it, or if you knew anything about it.
I think the opinion was what I give it. The homogenization. I don't think anybody's doing it in bad intention. Not to put any spores in there or anything, but it was just that if each company had a mycologist or a technician that is well experienced doing spore extractions and quantification, and have more power decision making above the sales people, they have a better product. Because if I'm in quality control and my hierarchy is on top, I'm going to call the shots, and I'm going to say, this product is not good because we checked it. It's not good. Don't sell it. It's not good. Send it back. Instead, a lot of the quality control is at the bottom, so the salespeople are on top. And so the salespeople will sell the product regardless if the product is not looking good. So sell it, sell it, sell it. And so to me, QC had to be on top.
Yeah. Well, there were some brands out there that had literally zero. It was fancy water. So
yeah, geez,
it's eye-opening.
Yeah, but the ODA also was doing bacteria, and I don't do bacteria.
All right, our next question is from Sean Russell. How far will the hyphae travel to make a connection with another root or hyphae pair?
Oh man, it can be meters. Meters. Yeah.
Can you go a little deeper in with that, or?
Well, I mean, as I said, imagine a corn root system, and they're all patchy, and the fungi can go straight, out into the soil. It's hard to say, but it can be. I mean, as long as they're attached to the root, they just can grow. They just grow radially. If you look at a three-dimensional one, you might have small circles. Small spheres of mycelia, or bigger circles of mycelia. Some can be very big. Now, remember, in the natural soils or outside, species have a different function. I mean, it might be some redundancy. What's the point of having 6, 7 species in a mycorrhizal product? Why would you want to apply 6 or 7 species? What's the reason behind ecologically? Well, maybe I'm trying to justify. Well, maybe one species is better than the other to uptake phosphorus. Maybe one is more tolerant to salt. And so you got a diversity, assuming they're all going to grow and attach to the root. In theory, that's what they're doing. They're saying diversity of things, not just one species. So that's one way to look at it. And the soil, the same thing. Not all species are going to colonize at the same rate. They're not. Some can overlap, and some are all over the place. And so it's a jungle out there. Everybody's looking for space, and the species they're competing. On the root system. The soil, natural soil, is one way. It's very complex, but when you put plants in a pot, the system is a lot simpler. And so, you can manipulate things, but it's not the same when you work with natural soils. Totally different.
Yeah, well said. This is Alex Steeker. I compost using worm bins. Is the liquid discharge from the bins a good way to help promote fungal activity? I've read a lot of mixed info on using the liquid. However, my personal experience using it, I've always had good results and have noticed mushrooms often follow its application.
Totally different thing. The mushrooms are very likely to be decomposers. They're not going to be mycorrhizal because there's no mycorrhizal fungi that attach to Cannabis mycorrhizae way and produce mushrooms. They're only release spores in the soil. Remember the Arbuscular? They produce the spores and leave them in the soil. They not produce mushroom. Those decomposers that come from a different, they either on the substrate or they're coming from somewhere. The main thing, it's hard for me to say, use this formula, this liquid and that. To me, at the end, is what's the phosphorus content in your amendment? Because, if you're going to have high phosphorus or high nutrient levels, it's very likely that the fungi, if you want to check the mycorrhizal fungi, may not do well because it may be too rich.
It sees it as a pathogen, right? Like if it's too easy to get it, it will repel it because it's using too much photosynthesis throughout the day.
Well, no, the mycorrhizal fungi, it may germinate, but it may not do well. Let's assume it connects to the roots, but it may not do well if the system is too rich. If it's high phosphorus, right, you see very little colonization, if any. The same with nitrogen. And I have some data we had with Leighton a couple of years ago.
Because the system's too lazy, right? It's too available. So why would it need to put up that?
Yeah, it's too available. And so it's amazing, all the cannabis growers have their systems. They got their own recipes, and they change it every time. It's fun because things go quickly, but it's hard to keep track of each organ, of groups of organisms they're doing in my system. In the next year, you change a little bit different for whatever reason, and so. If you don't have data, consistent data, we're all living on assumptions. Here's another thing that I wanted to make sure I mentioned. I give a talk in Portland to cannabis growers. I can't remember, a couple of years ago, maybe 3 years ago, and also in Humboldt, Garberville. And I asked the question: raise your hand, those that use mycorrhizal inoculants in your system. And pretty much the whole group raised their hand. I said, now, how many of you actually, at the end of your cycle, check for mycorrhizal colonization on the roots where you applied them. Forget about controls. Just see if the inoculant works. Zero.
Yeah.
And so if they say my plants look great, they do awesome with this product. Now, what's in that product? Did they have a little fertilizer? Did they have a little organic matter? Did they have bacteria? Did they have mycorrhizal fungi? That's what the label says. But that doesn't mean that's the whole content. If you were to run pyrosequencing on those things, you probably have a bigger list of organisms in that product than the ones they're saying in the label.
Mm-hmm.
Those are the ones that they produce and they add to your package or your container. But there's more things. If they're not sterile, there could be more things in there. You just don't know. On top of the ones that come with the soil that you bought at the store or that you made. So you have a medley of, a big mess of stuff in there. And so when you put your plant, you don't know which one is causing the effect. To know that statistically with the scientific method, you need to set up individual treatments with individual organisms on top of combinations of organisms to see if there's synergistic effects or confounding effects and there's replications and variants and is it significant, is it no? And then at the end, it says, okay, which organism is the one that caused the effect? What species of mycorrhizal fungi actually grew? Because I put 4, but I'm assuming that the 4 work at the same percentage or at the same rate. It may not be the truth. So we're assuming a lot of things because we don't check it. We're not cannabis growers are not in the business to check if mycorrhizae is growing. They're in the business of producing, flowers or quality of the terpene. They don't have time because it's time to throw this away or recycle it or whatever and set up again because the season is coming. And so there's no budget to keep doing that. It's not research. And so there's a lot of things. And a lot of times we only do 1 sample or 2 samples. But you got 100 plants. And so your data is based on 1 sample. And so are you going to manage your soil based on 1 sample? And that sample is because this one in the corner, because it was closer to get to. I don't want to walk to the other side of the room to get the sample. And so you're not sampling representative, you're just sampling out of commodity or comfort. And so sampling is usually overlooked when we try to assess things. And sampling to obtain data is expensive for anything. And so the grower is not in the business of getting research data unless it's a research. And so not everybody has the budget to do a lot of deep, and careful examination. You got to know the limitations of your data. At one time, and this goes to, we still have time. Many years ago, this is not a cannabis, a truffle grower, I'm not going to say name, a truffle grower, wanted to know if his trees inoculated 10, 15 years ago with truffles from France. Wanted to know if it's still active. So he lost track of that truffle grow, that orchard. This is where like hazelnuts, filbert trees. So I was talking a lot about sampling. I said, well, how many trees do you have? This is a consulting job I did 20 years ago. He said, I got 50,000 trees. 50,000 trees. They were about 15 years old at that time. Inoculated with the French truffle. They were they're inoculated, right? They did inoculation. They grow in the soil. They did that stuff. I said, well, how many samples are you going to send me? He said, well, I'm going to send you, I don't know. He just came up. He sent me 24 samples of roots. And just go back to the corn example. You have a whole tree of roots. You just imagine the system of a filbert tree. And you send me a handful of roots. And in the patchy distribution of the truffle individuals in the soil, it can be anywhere. It is not going to be 100%. So there's a possibility that I may not catch the fungus when he sampled the roots. So he sent me a long root thing with tips. I can't look at the whole thing, so I have to subsample and get 1,000 root tips, 1,000 in a hand. I mean, it can be overwhelming. Just imagine the thousands of root tips in the soil. Now, I did not find the truffle based on morphology on any of those samples. What's the critical thing here? As a consultant, I said I did not see the ectomycorrhizal fungi distinguished by the truffle. The characteristics are not there. But I'm only responsible for the subsample that I did. I had no clue what the rest of the tree for that individual is. I'm not responsible for what was not on the sample. And I'm only responsible for 24 samples that came from 24 trees. About the 49,976, I'm not responsible for those. And I was very clear because I didn't do those. I only do that tiny subsample. Just think about it, 50,000 trees. So I'm not going to be responsible for what you do. Well, he went walk away. It's sad. Guess what he did? He chopped the whole orchard. Completely.
Why?
I don't know. He just said, no truffles here. I said, remember what I said? I'm only responsible for 24 samples. It's your thing. He logged the whole freaking thing and sell firewood or something. I don't know what he did.
Oh my God.
But, I mean, that's it. It's like, so sampling, it's something to keep in mind, the limitation of your data. Don't make big soil management decisions based on very little data. Because, you took on an acre or 10 acres, but the data comes from a cup of soil. You're assuming that the rest of the stuff is the same thing. When
you're getting your soil samples in, like what grade would you give most people? Like you see a variety of things, and I know Leighton is one of your biggest cheerleaders.
Yeah, we'll see more. I'll advance to make sure I cover the rest of the stuff. But I'll touch it up.
Okay.
You can save. Maybe I'll save 10 minutes for more questions. Let me just keep going. I caused a lot of controversy now in the audience. I'm not looking at the chat, so.
Yeah, it's hard to keep up with the chat, and I've never seen somebody put a tamale up there for no reason. So appreciate that.
Well, it's just trying to make you hungry, make you guys hungry. So, mycorrhizal fungi in agricultural systems, the challenges, the crop production. We want us to be sustainable with low chemical inputs. The uptake of nutrients needs to be top notch so you can have good yields, long-term sustainability. And you're looking for the best group of microbial organisms, the holy grail. Everybody's looking for the best of the best things, bacteria, worm, everything, We want a miracle formula. The arbuscular mycorrhizal fungi, as we know, they play a vital role on the uptake of water, nutrients. They suppress soil pathogens, and we're just looking for a better plant development. And so that's the highlight of the mycorrhizal fungi for anybody to study. Just quickly, when we talk about beneficial organisms, the miracle formula we're looking is that we want soil structure. If you're in the soil system, natural soil system, you want to have access in the soil sources, but you don't want to deplete them. You want to prevent disease. You want to reduce the stress of the plants. You want to increase plant performance, better terpenes, a lot of THC, a lot of CBD, and you don't want to spend too much. So it's the budget is not there, so it's not easy. This one is just to represent, some of the things about the distance. Plants on the left is basically a regular plant or grass, and then you get the clovers. Some plants do different things, nitrogen fixers. Some create better aggregates, some create better mycorrhizal colonization. Companion plants, we can talk about companion plants too on the question section. Talking about mycorrhizal inoculants, some are seed coated, some are powder, some are root dips, some are granular, some are liquid. The bottom right picture is all the yellow, kind of a beige brown color, coffee color. Spores and root fragments, the whole thing that's produced in vitro. That's thousands and thousands and thousands of spores. The one on the left is stained spores, right next to a vermiculite, and a little root fragment on the right, on the top. So the mycorrhizal inoculation, it's different forms, depends on what you got. So you can put the powder on a sprayer, a water buffalo. You put it in a compost tea and mix it all up. You put it in a sprayer. We're doing inoculating this. The one on the right, we're inoculating a brand new sports field for an NFL team. And you can see the brown shade area on the right as the spores. The inoculant changes the shade. This is before they put the sod on top. And we monitored that the whole year. It's pretty cool. So mycorrhizae on an NFL team. Then we did another inoculations on San Luis Arch, the state park. And then also we're working at Expedia campus in Seattle. These are the typical pictures of big plant, little plant, mycorrhizal, no mycorrhizal. I get tired of those but I need to show them to you. They're pretty old pictures, Potatoes, root biomass, mycorrhizal, grapes. Grapes are good for mycorrhizae, the low and this is just an image. I can be lying to you, but this is what they told me, it can be a total different time of the year, But anyway, so I just take it as a right statement. Non-mycorrhizal versus mycorrhizal. But the message for me here is more than just big plant, little plant. They're so complex. The systems are so complex. There's so many functions, interactions, organisms in soil experiments that a lot of times the controls are not set up correctly. The data is not solid. Sometimes the experiment worked really well, and sometimes the experiment didn't work really well. The data was kind of not there. But I learned in the graduate school. I got my PhD 30 years ago at Oregon State University, and I learned from some of the committee members that never repeat a successful experiment. So, if you want to produce, publish a good data, never repeat a successful experiment. Anyway, so mycorrhizal fungi, their assessment is one of the measures used to determine soil health, and this is what we're doing in many projects throughout the U.S. The picture on the left, what I'm sampling, this is plain London trees in the St. Louis State Park. The plants around the tree, all those brownish, these were volunteer weed plantains. Now, immediately when I saw this, the INBAM Institute keep this culture collection of mycorrhizal fungi in vivo. What plant do they use to keep the culture collection? They use plantain. They use another grass, some bracharia grass. And so, to me, you put that as plantain. What is plantain doing in this disturbed soil? They're tapping into the mycorrhizal fungi. And so they're keeping the mycorrhizal fungi active. And so from the ecological point of view, I said, these plantains are good. The companion plants on this tree are awesome because they're just stimulating the mycorrhizal activity. Being a little islands, each root, each plant is being a little island of mycorrhizal fungi. But what happens? The landscaper comes and start chopping them away, taking them apart. I said, well, it's good for aesthetically, but below ground is a different story. So, in the middle picture, in the Jackson Park in Chicago, we're looking for hot spots. So we go to a plant community and we start looking, vegetation, spore assessments, and so we're trying to find what area of this park or this plant community, this urban community, where's the mycorrhizal fungi being really productive based on spores and colonizations. So what are we trying to do this? Well, once we identify and that this area is going to be disturbed and build another new structure, we're trying to save the legacy by preserving the region already adapted to those conditions. And the bottom picture is the London Plains in the state park in St. Louis, the arch. We put about, well, I didn't, but we assessed, we inoculated 800 London plane trees in Asian. Excellent numbers we got on that. So how do we determine the presence of mycorrhizal fungi in the soil? Root colonization in the roots, by clearing and staining the roots and looking under the microscope, dissecting scope, and then confirming them in the compound if you have to. But I don't do that often anymore. So you can see where they are. The other sucrose, sugar levels, solution to extract them, and then we do the examination. Others are molecular analysis or doing soil biology. That's that we do. The clearing and the staining, and depends on what you're looking at, you go to the microscope. The clearing and staining we use or estimate the whole root length of the roots, and then I estimate the colonization in every piece that I see. And then the microscopic examination, if you're not familiar with the mycorrhizae, you keep going back to the compound scope, but it takes too much time.